want to take notes.
You look better, Courtney. You must be feeling better.
Whoa.
You can tell.
Did she look like crap?
She pretty much did all last week, yes. Well, good. No, it's fine. I'm noticing a lot of my students are sick.
It's okay. Mr. Bell called me 4 times. My little brother threw up 15 times.
That's terrible. Okay, guys. Hey, last Thursday, remember last Friday we isolated DNA from a strawberry, just for fun, just so we could say we did. Last Thursday, I actually reviewed a process called DNA replication with you. And remember, just a quick little summary. All we were doing was taking one piece of DNA and making another copy of it, an exact duplicate copy. Okay, remember, pretty much it involved you just splitting DNA down the middle like a zipper, and then an enzyme called DNA polymerase coming in and just tacking on new nucleotides. Okay, it was a real simple process, more or less. Today, I want you to kind of keep in mind this is a totally different process I'm talking about today, but it sounds a little similar to what you've already learned about, but you really have to keep it separate. Before I go into that process, though, it's a good time to review something we learned many chapters ago. I don't want you to write this. You already have this in your notes. Shh. You already have this in your notes. Many chapters ago, we learned about parts of a cell. And you're going to need to know these in a couple weeks anyway when you guys have your midterm in here, okay? So anyway, parts of a cell. Now, I'm going to draw just a typical eukaryotic cell, and you guys should know that if it's a eukaryotic cell, what does it have?
Nucleus.
A nucleus, okay? And inside that nucleus is what?
DNA.
Piece of DNA. Okay, that's good. All right. Good. Now, another thing we learned when we were going over parts of the cells, we said that DNA is kind of selfish in a way. It's kind of like the boss man. He runs the show. But he never leaves the nucleus in terms of a eukaryotic cell. Never leaves the nucleus. He stays there. Okay, so another thing we covered was the fact there's this other organelle. I'm going to just start drawing organelles and make sure you guys kind of try to remember what these things are good for. There was another organelle just outside the nucleus that sometimes had these tiny little dots on them and sometimes did not, in a rough form and in a smooth form. Number one, what are these little dots that I'm drawing representing?
DNA replication.
I don't think I heard it. R.
NA.
I.
Ribosome.
Ribosomes. Okay. Remember, way back many chapters ago, when you learned about what a ribosome does, you should have learned or memorized that these are the sites of protein synthesis. In other words, this is where protein's made. Okay, the ribosome is the site of protein synthesis. In other words, the ribosomes are where protein is made. And if you remember, how exactly that happens is this piece of DNA actually gets converted into RNA, and then that RNA actually leaves the nucleus, hooks up to one of these ribosomes, and literally then this RNA is converted into a protein. And I'm just going to represent the protein by a blue blob there. So let me rehash that. You have DNA in the nucleus. It's going to get changed over into RNA. That RNA is going to leave the nucleus, hook up to one of these ribosomes, and out's going to pump a protein. That is what I'm going to talk to you about in the next couple of days, are the details of that, going from DNA to RNA and from RNA to protein. That is called protein synthesis. That's basically, again, what this next kind of the deal talks about. We're not taking DNA and making a copy of it. We're taking DNA, making an RNA, then making a protein. Okay, well, while I have this up here, the reason I bring that up is because, you know, we learned just kind of a review. That protein that was just made is going to take a little ride. That protein's going to take a ride in a little thing called a vesicle. which is just a little transportation vehicle thing. I like to think of vehicle vesicle. This thing's going to make a pit stop before it leaves the cell to another organelle that you guys should know about. This thing kind of looks like the endoplasmic reticulum. By the way, that's what this thing is. I don't think I said that word. Remember, this is the endoplasmic reticulum. Oops. Endoplasmic reticulum. Plasmic. I'm going to abbreviate here. Endoplasmic reticulum. Sometimes the endoplasmic reticulum is smooth. Sometimes it's rough, depending on if it has those ribosomes or not. Anyway, going back here, we just made ourselves a protein. That protein's going to take a ride in a vesicle. It's going to make a pit stop to another organelle, which is going to kind of fine-tune it. Okay? It's going to modify it. Add a little sugar to it if it needs it. Add a little phosphate group if it needs to it. It's like the UPS center. What's that organelle? Golgi. Remember, the Golgi apparatus, or Golgi body, whatever you want to call it, is an organelle that's going to receive this protein and fine-tune it. Like I said, usually what happens here, that protein blob is going to get a little addition onto it. Maybe a little sugar, which I'll represent by pink. Just a little sugar added to it. Now that protein is ready to be shipped out. Okay, so it's going to hop on board another vesicle, and it's most likely going to leave the cell. Sometimes, though, it won't. Sometimes proteins will be made that stay in the cell to form other structures. Name me the structure that is like the cleaner upper of the cell. It's another organelle in here. Lysosomes. Remember lysol? Lysosomes, these structures are filled with these like enzymes that break things down, like Lysol breaks things down, cleans things up. So some of those proteins that this cell is making is actually going to go on to form these enzymes of lysosomes. Another structure. Name me the structure that's kind of like the scaffolding in the cell. It kind of runs all throughout the cell, being kind of, it's like gives it its shape. That's called the cytoskeleton. Easy to remember. So that thing's made up of proteins. So some of these proteins that you've just made are going to stay in the cell and form the cytoskeleton. Boy, I tell you, all this work going on sure requires a lot of energy. Name the organelle that's going to power these reactions. Mitochondria. Remember, mitochondria look like this. Have a double membrane. Mitochondria are making energy. By the way, what's the form of energy that a cell uses? It's not gasoline. Our mitochondria better not be pumping out gasoline. I'll tell you what, our cells wouldn't like that. What kind of energy source do our cells do like?
Sugar. Does it start with a C?
Sugar goes in. So glucose goes in. What pumps out? I have a jar that I made of it.
Phosphate?
Phosphate.
No, that's just part of that molecule, but that's not actually phosphate.
ATP.
ATP. Remember, ATP is like gasoline for the cell, but it's not gasoline. Like I said, real gasoline would not be good. But ATP and gasoline have something in common. They're both really huge molecules with lots of bonds, joining lots of atoms. It is a powerhouse of energy in those bonds. So I guess in a way you could say ATP and gasoline are similar, but they are definitely different in a lot of ways too. Okay, I realize this is a mess, but we do need to remember our parts of the cell. I'm trying to think of anything else that I'm forgetting. I'm sure I am. I didn't really talk about the plasma membrane. Oh, if this was a plant cell, what two other things would be here?
Cell wall.
Cell wall, which would be out here, and what?
Cytoplasm.
Nope, cytoplasm means both, but it does start with a C. Chloroplasts. Remember chloroplasts. Chloroplasts are those organelles only found in plants, is where photosynthesis occurs. Obviously, our cells don't have them, kind of a deal. Okay, so I know this is a mess, but take some time, guys, to kind of go back over those organelles. All right, moving along. Now let's get specific to what I wanted to talk about with you guys with this chapter. The whole point of me doing this, guys, was number one, to review your parts of the cell, but then also to tie in the fact that the big thing we're going to be talking about now is related to some of the parts of the cell. No, I just don't feel like using it today. Kind of in the blackboard mood. So, but it is. Okay. Hey, this next thing I'm about to write up on the board is probably one of the most important things I will write the entire year. If my board ever dries, I think I'm just going to keep going anyway. And actually, this is something you did see with Mr. Sisson. Wow, this is really some powerful stuff up here. DNA goes to RNA, goes to protein. Okay, big picture here. Guys, when you guys look into a mirror every morning or evening or whenever you look at yourself in the mirror, okay, you are seeing a lot of protein. That's what we're made up of. That's what all living organisms are pretty much made up of. Skin, hair, organs, blood. Your blood is protein. All the enzymes that keep you going are proteins. Bone, protein. Everything about you is pretty much a protein. There's some other stuff too, but really, we are big bags of proteins. You should all be writing this down. I want notebooks in front of everybody, please. Okay. We're big bags of protein. Well, I don't know about you guys, but let's look at this from a logical standpoint. If we're made up of protein, we better find a way to make the protein. And to keep making the protein while we're still alive. Okay? That is what this next process is about. But this next process I'm going to talk about is broken up into two parts. So it's a two-part process. It all starts with your DNA, though. Now, at this point, you guys should be pretty comfortable with the molecule of DNA. You guys have made a model of it with paper and cut it and scissors and all that. Some of you guys even made me some 3D models, again, which are extra credit between now and December 21st. You can make me a 3D model of DNA like you see around the room. So you guys are actually comfortable with the molecule of DNA. Please understand, in every one of your cells in your body that make you up, you have a piece of DNA in there. Okay, and you have a piece of DNA in every cell in your body, and it's the same piece of DNA found in every cell of your body. Now, your DNA is the same as the person sitting next to you by about 99.9%. It's just that 0.1% that makes you look a little different, be susceptible to maybe some disorders, whatever, that makes you a little different. 99.9% similar to the person sitting next to you. Your DNA is the same. Scary thought. Okay, so the reason you have a piece of DNA in every one of your cells, though, is that DNA is going to provide the code. It's going to provide the code that's going to go on to make a bunch of protein. Remember, every one of your cells, as we saw earlier, is like a big factory, a protein pumping factory. It's basically making a bunch of protein. The process starts with your DNA. Your DNA has to, which provides a code, which is going to be the code to make that protein. All right. The name of it's a two-part process, like I said, because it doesn't just go right from DNA to protein. Why? Because DNA doesn't even leave the nucleus. Remember, it's like a big, old, lazy boss who just sits in the nucleus and doesn't go anywhere. So DNA pretty much hires out a messenger. That's the job of RNA. The name of the process where DNA gets changed into RNA is a process called transcription. Okay? Oops. Thank you. Transcription. All right? This process occurs in the nucleus. Again, because remember, DNA doesn't leave the nucleus, so this process takes place in the nucleus. And, yeah, I think I'll leave it at that. Okay. But you're not done yet. All you've done at this point is just make a piece of RNA. That RNA now is actually going to be another code just a slightly different one from the DNA. But then that code also needs to get changed to an actual protein. The name of the process where that takes place is what? Translation. Okay. You do need to remember those two names. This is easy. It's in alphabetical order. Transcription C comes before translation L. So transcription always comes first. Like if you look at it in alphabetical order, transcription comes before translation alphabetically. Okay, anyway, translation doesn't occur in the nucleus. This actual process is going to take place on a ribosome. It makes sense. Remember, a ribosome is the site of protein synthesis. So it makes sense that this last part of this process where you're actually going to be making the protein occurs on the ribosome. Boy, is this important.
It is.
It really is. And I mean, look at it this way, guys. You know, when you're sitting here, your DNA, the code that's in your DNA is pumping out protein, and it's doing it in this manner. Okay, this whole process is called protein synthesis. It's a two-part process. But this is the way you are able to generate all these proteins that are going to go on to form your skin, your blood, your bone, your organs, your everything about you. Everything about you. So you better have a way of doing that. So, questions on this? This is kind of like an overview. All right. A couple more details I want to bring up. I'm just going to use the eraser so it can dry. Now let's go a couple other details. That's not even working well, is it? Details to kind of talk about before we get to the hard details of transcription and translation. I do want to talk about the major differences between DNA and RNA. Obviously, you guys remember the first step is taking DNA and converting it into RNA. We better figure out what the difference between these two molecules are. Good news is they're very similar. They are both, and you've seen this before, they are both made up of these units called nucleotides. Both of them. So is RNA. So nucleotides make up DNA, nucleotides make up RNA. Remember the circle represented a phosphate. This phosphate is the same as that phosphate down there. This little house represented the sugar. In DNA, it's called deoxyribose. That's where the D in DNA comes from. In RNA, it's called ribose. That's where the R in RNA comes from, the name of the sugar. Okay. The other part is this base, this nitrogen base, and you guys already know this. In DNA, that base can be either an A, T, C, or G. You guys at this point also know that in RNA, it's almost the same, but what's different? Yep. You don't have a T, you have a U instead. So if you're talking about RNA, you will never see a thymine. You'll never see a T. Another difference between these two molecules is this. DNA, as you guys knew from your models, has two strands, two sides of a ladder. And I'm filling them in with those steps. RNA, on the other hand, only has one. It's almost like a non-working ladder. It's like half of a ladder. So here's the deal. On these pictures you made, you know, you guys made two sides of a ladder here. That's DNA. But RNA would basically look like half of the ladder. It's only one strand. Always. So, all right. The only thing I want to talk about then is this. There are actually three types of RNA. Who can name me one?
Messenger RNA.
Messenger RNA. Very good. Which is abbreviated mRNA. See, they make this easy. The M stands for messenger. The job of messenger